Okay, the chemical messenger will be received by receptor. ये रिसेप्टर क्या करेगा, मैसेज को लेगा एंड देन इट विल ट्रांसफर टू द सेल, सो अगर हमको ड्रग और मेडिसिन इन बॉडी में किसी बीमारी को दूर करना है, सो हाउ इज इट पॉसिबल, हाउ द ड्रग वर्क, हाउ द एंजाइम work, we will discuss in the next slide. So, how the enzyme work? Suppose this is an enzyme, right? There will be one active site. The shape of the active site will be similar to the shape of the substrate. Okay. So, when the reaction takes place, you can see the shape of this enzyme and the substrate is same, so it gets bind over here. For example, think about it. This substrate is some harmful chemical. Right? And this enzyme gets interacted to the substrate and they make a enzyme substrate complex. You may think how this substrate get bind to the enzyme. In our earlier slide, I have shown you that enzymes have hydrogen, oxygen, all this kind of elements. They can get attached, they means the substrate can get attached to the enzyme by hydrogen bond, ionic bond, dipole-dipole interaction, van der Waals force of attraction. All this kind of interaction can be possible for the interaction of enzyme and the substrate. When the enzyme and substrate gets combined, some product is formed, and after that they may or may not lose the interactive site, or we can say the product, after the formation of product, the substrate which is no more left, the new product is formed, which earlier was not possible. After the interaction with the enzyme, then only the formation of product takes place, right? So if it is something harmful, we have to break it, right? Here comes the role of a drug. What does the drug do? What we can do? Think this we can wait for a second. We can destroy this site. If this substrate is harmful, either we can destroy this site, or other option, we can fit something else in that site so that the space will be occupied and the substrate will not be able to get into that place. So two options are there. One, we have to fill this place with something, and second, we have to destroy this site so that the substrate doesn't form complex with the enzyme. In our next slide, we will discuss these two factors. Here you can see when the reaction is taking place, this is the enzyme, right? And this is the substrate. The shape of the active site and the substrate is similar. So formation of enzyme substrate complex takes place. Somehow, if we destroy or block this active site, then this complex will not form, and if this complex is not formed, then we can easily get cure, right? Here comes the role of the drug. This is a drug. It has similar shape with that of the substrate. Then the active site of the enzyme get attached to the drug. Okay, when the drug forms a complex with the enzyme, then that active site is blocked. So now the substrate cannot enter. The substrate which was causing the human tissue harm is no longer available for formation of complex with the enzyme because the active site of the enzyme is blocked. This is one of the way to stop the substrate to enter the active site and harm human tissue. Here you can see the enzyme has been inhibited. The substrate which was supposed to enter the enzyme, or we can say the substrate which was supposed to form the enzyme substrate complex, is no longer available because the drug inhibited that active site. So this process is known as enzyme inhibitor. Next we will go for the other thing that I have told in previous slide, that if we destroy the active site, think, this process is known as allosteric inhibition. Allosteric inhibition. What happens in this? Suppose this is the active site. Suppose this is the active site of the enzyme. Here the substrate is supposed to come, but there is one allosteric site where the drug enters. This is drug or medicine. This is drug or medicine. When the drug enters the allosteric site, it changes the shape of the active site. When the shape is being changed in the active site, the substrate cannot enter the active site and form the complex. In these two methods, we can stop the disease by these are the two ways in which we can stop the interaction of a substrate with an enzyme. This is drug enzyme interaction. In our next class or our next video, we will discuss about receptor target interaction. Okay, that's all. Thank you.